Hello, my name is Dolors Villegas. I work at IRTA in Spain, and today I will explain the surveys of alternate and auxiliary hosts of stem and yellow rust pathogens in Spain. This is the outline of my presentation. We will start with an introduction, followed by a section of field methods and observations. Then I will explain methods and results from molecular analysis, and we will finish with conclusions. Spain is located in the south of Europe and could serve as a potential bridge for cereal rust movement between Africa and Europe. There are three subspecies of Berberis vulgaris in Spain: vulgaris in the northwest, Seroi in the northeast, and Australis in the south, also known as Berberis hispanica. Of these, the Seroi and the Australis subspecies are indigenous to Spain. Both, in theory, could act as alternate hosts for wheat stem rust and stripe rust pathogens. Wheat in Spain occupies around 2 million hectares. In the areas marked in blue in this map, there is more than 90% of the surface devoted to wheat cultivation. The objective of this study was to test if the barberry subspecies indigenous to Spain effectively act as alternate host to Coccinia infecting winter cereals. For this study, barberry populations, wild grasses from the Poaceae family, and cereal crops were surveyed for rust infections in several provinces of Spain since 2015, but more intensively in 2018 and 19. In April 2018, before the BGIR conference in Morocco, we made a survey trip in several provinces of Spain to locate and sample barberry of subspecies Seroi and Australis. We could not find plants with infected leaves by that time, but some areas were visited and sampled later in the season. In 2019, we sampled those areas again and expanded the surveys. Samples for analysis were taken from the barberries, dried at room temperature, and placed in glassine bags. They were packed with special envelopes and containers and shipped with a special permit. Samples were sent to the Global Rust Reference Center in Denmark and the Cereal Disease Lab in the United States, and this latter required also a special label. All this process has to be perfectly coordinated with the lab receiving the samples. The results of our field observations showed that in northern Spain, Berberis vulgaris subspecies Seroi grows at medium altitudes, around 500 meters above the sea level upwards. Occasionally, it may be found in close proximity to cereal crops and grasses along fields, as shown in these pictures. The normal sequence of events is as follows. In spring, after leaf emission, the infection of rust in barberry, indicated in red in this graph, takes place in coincidence with flowering. After a period of active Aeschia sporulation, the Aeschia get dry and the infection in barberry finishes. Grasses become infected after this moment, and the infection is long because there are many species with differences in phenology. New and healthy branches grow in barberries meanwhile. Wheat and other crops may become infected some years depending on the environmental conditions, indicated here by mixed red and green, more or less at the same time than the grasses. As crops mature and are harvested in summer, stem rust survives in grasses. The grasses infected with stem rust we collected included Aegilops ventricosa, Avena steralis, Dactylis glomerata, some species of Elymus, Electrotrichum bromoides, and Ordium murinum, among others. In the north of Spain, stem rust infections on crops occur occasionally, while yellow rust infections are usual. However, in 2018, there was a strong infection in rye and wheat in some specific locations in Teruel province, where barberry was present and yield was reduced in more than 50%. In 2019, stem rust infections were observed also in Huesca province, but only in close proximity of barberry specimens and with low impact on yield. Those infections close to barberry proved to be rare and diverse. We also observed infections in Lleida province, where the barberry is not found, but the races found there were already described elsewhere. More information about the races infecting wheats may be found in another oral presentation by Dr. Patpour and also two posters that are presented in this workshop. In the south of Spain, Berberis vulgaris subspecies australis grows at high altitude, from 1,000 to 2,500 meters, and it is rarely found close to cereal crops. In the south, grasses and crops may become infected in spring, while barberry acacia appear in the summer. Summer nurseries under irrigation usually are infected with stem rust, but races found in these nurseries have also been previously described. At Cereal Disease Lab, two types of molecular approach were used to identify Puccinia from acacial samples from barberry. First was real-time PCR assay to identify Puccinia graminis and Streiformis, and the second was an ITS amplification, cloning, and sequencing. For the real-time PCR assay, we extract DNA from 3 randomly selected individual acacial clusters per sample. Real-time PCR assay was performed using one probe specific to Puccinia graminis and one probe specific for Puccinia streiformis, as described in Barnes and Zappel. The results are shown in this table. Of 22 samples received in 2018, 24 samples were confirmed as Puccinia graminis, and 0 were Puccinia streiformis. Of 29 samples received in 2019, 29 subsamples were Puccinia graminis, and none was Puccinia streiformis. For many other subsamples, the poor quality of the DNA made it impossible to perform the analysis. In the second method of analysis, the ITS region was amplified with ITS1F and Rust1 primers. 8 clones per sample were cloned and bidirectionally sequenced. Phylogenetic analysis using neighbor joining method was performed using 33 reference sequences, including Puccinia graminis from wheat, rye, oat, white grasses, Puccinia streiformis, Puccinia arenateri, Puccinia brachypodi, and Camisella. These reference isolates were selected because barberry can host all these rusts. A 1.2 kilobase fragment containing the region of interest was successfully amplified from 36 samples. 35 of these samples were identified as Puccinia graminis complex. 24 of them grouped with Puccinia graminis from wheat, rye, and wild rye, marked here in red. 11 grouped with Puccinia graminis from oat and other wild grasses, marked here in green. And 1 sample grouped with the Puccinia brachypodi species complex, marked here in pink. From the acacial samples collected in Spain during 2019, unique and significant virulence combinations were detected. The Cereal Disease Lab derived 190 isolates from 12 samples and identified 148 unique races. A high frequency of virulence to SR24 and SR31 was found, either alone or in combination. The virulence to UG99 effective genes identified include all the list of genes that you can see on the screen. In conclusion, we can say that the acacial infections in Berberis vulgaris, subspecies Heroi, and australis were readily observed at several surveyed sites in Spain. In the north of Spain, barberry grows in proximity with grasses and crops that are infected with stem rust following infections on barberry. Race analysis of stem rust isolates derived from acacia and barberry, as well as field observations, indicated that the sexual cycle of Puccinia graminis is active in Spain and contributes to pathogen variations. We thank the Delivering Genetic Gain in Wheat project, the Cornell University, and the Polar Global Rust Initiative for their support, and also thank to all of you for your attention.